slowly streaming in here from the last session. So maybe just give people a minute while Marnie gets her screen shared, and I'll pass it over to Maddie.
Thanks, Mackenzie. Yeah, hi everybody. Happy to be here moderating our forest pest session. Just wanted to remind everyone that there will be time for questions after each presentation. So if you have questions at any time, please pop them in the Q&A box and I'll read them to our speakers at the end of each talk. So with that, let's get started with our first speaker. First up, we have Marnie Demand from the City of London to talk about long-term monitoring in park woodlands. Thank you so much for being here, Marnie.
Hi, thanks for having me. I'll get started then. So I'm talking about the establishment of permanent sample plots in the City of London's woodlands. So why we did this is basically to meet some...
I'm not seeing your slides. I don't think you shared your screen.
Not sharing now?
Perfect. Yeah, that's great. Thank you.
Great. Okay, so starting over then. I'm going to be talking about permanent sample plots in the city woodlands that we've set up in the City of London. So as far as why we're doing this is to meet policy objectives, but also to expand our knowledge base and to ensure that we're ready in the event of more invasive species arriving, and also to quantify carbon storage, which will be in compliance with one of our objectives of the Climate Emergency Action Plan. So that ties back into our policy objectives. So I'm going to go over why we're doing this, that's part of it, how they've been installed, a few of our results, and then how they might help us in the future. So one of the main reasons we're doing this is because the previous information we had on the city woodlands was quite limited. We know that we've got approximately 290 woodlands. We know where they are. We know the area of each one. We've got a shapefile with their locations on it. And they're all classified based off of the type of park that they're in. There's a list of park types on the slide, including woodland park. However, according to the information we have in our system, only 17 of these woodlands are in woodland parks. So we know that we need more information. We also know a few of the trees that are within these woodlands when they're in woodlands that also happen to be manicured parks, which is kind of counterintuitive. But some of the areas classified as woodlands are not as woodland-like when we get out there. So that's why we're doing it, is to enhance our understanding of what we have. One of the things that has really clarified the need for this is we recently had an outbreak of beech leaf disease in the City of London. It was found first in 2023, and it's believed to be caused by a nematode. I'm not sure if everyone's aware of it currently, but the most characteristic symptom of it is this striping of the leaves between the veins. So that was detected again first in London in 2023. It's new to North America. So we had a contractor go out in 2023 and look at some of our woodlands to determine the prevalence of beech leaf disease within the woodlands, as well as our city street trees that are beech. There are not a lot of those, but they looked at those as well. So they looked at 47 woodlands. Of these, 29 had beech, and this is a result of the trees that were infected versus not infected. About 70% of the trees had beech leaf disease. As of 2024, when I was installing permanent sample plots last year, all of the woodlands that had beech leaf disease detected in them. Not necessarily every tree appeared to be infected, but all of the woodlands with beech had at least one tree that was affected. So going back to the work that was done in 2023, if we had had more information on which woodlands had beech in advance, we could have been more effective in where we sent the contractors to look for beech leaf disease. So that sort of helps to clarify one of the benefits of this is you can really target your actions based off of what you have when you know what you have. In terms of how they were developed and the methodology for that, I reviewed methodology from a few different provinces in Canada. There wasn't any for Ontario, so I looked at Nova Scotia, Alberta, and British Columbia's methodology. These are for slightly different purposes than what the City of London was looking at. So it was revised a bit, and the plot size that was done for the permanent sample plots was 0.04 hectares, which was based on Norwalk et al. 2008, to get the best quality data we could while minimizing time required. They were still quite time consuming to inventory and sample. However, it was a balance between how much data we could get and how much we could get done. So in terms of the placement of the plots, I overlaid the City of London with a fishnet grid, which is just this grid pattern. In the middle of each grid box, there's a point random based on the middle of that. And then from there, one plot within each woodland was randomly selected from the points within the woodland. We only were able to do one permanent sample plot per woodland was our goal over the next about 3 years due to resources and how long they take and having 290 woodlands, which is actually a number that increases slightly every year. So that's how the locations were selected. Once we got out to these permanent sample plots, I collected attributes of the woodland, the permanent sample plots, and then quadrats located around the perimeter of the permanent sample plot. So if the box, the white box here is the park, you've got a woodland within the park, and then within that woodland is the permanent sample plot at one of those randomly picked points from the fishnet grid points. And then around the perimeter of it were 8 quadrats. Again, it's a 0.04 hectare plot. And then the quadrats were each 1 meter squared. So 1 meter by 1 meter. At the woodland level, we collected qualitative data only. So that would be a list of the tree species and shrub species present, categorization of the abundance of trees, shrubs, and herbaceous or ground cover plants that were invasive, as well as any wildlife observed, human influences on the site, for example, unauthorized trails that had been created, excess garbage, things like that, bike paths that were not supposed to be there. Basically unexpected, well, not unexpected, but human influences that were not intended, but were present just in terms of degradation and damage to the woodland. And then also a qualitative description of course woody debris on the site. This is an example of one of the permanent sample plots. My backpack marks plot center with a lot of vines present. So they were not all easy to get to. And I've got a few other pictures coming up of what some of the other ones looked like as well. So they were highly variable. Within the permanent sample plot itself, within the woodland, qualitative and quantitative data was collected, including a complete inventory of all trees over 10 centimeters in diameter at breast height. Any shrub species, if it was unclear whether they were trees or shrubs, basically anything woody that was over 10 centimeters in diameter was included in that tree inventory, regardless of whether or not it was like a buckthorn, which may be classified as a tree or a shrub. Also did some heights, including the maximum height of a tree within the plot, height of the understory layer, the amount of the plot vegetation that was at the understory or canopy level that was covered by vines, and the species, and then the site type, which was a description, for example, if it was a woodland in the middle of the woodland or an edge, or if it was a park type habitat. So that was a description of the site there, which fell into several categories. Basal area of the forest was also collected from plot center, so that's not necessarily restricted to the permanent sample plot because it is a variable area plot, but it was based on plot center. In some cases, it would be biased if the plot was on the edge of the woodland. Those plots that were on the edge of the woodlands were not excluded. We could have buffered the edges of the woodlands and not done any sampling where there would be potentially parts of the plot outside of the woodland or quadrats outside of the woodland, but we decided to include those points that were near an edge because a lot of our woodland is edge habitat, and it's important, we felt, to capture representative sampling of our woodlands, regardless of whether they were edge or middle of the woodland, proportional approximately to what we had. Within the quadrats, we collected soil samples, and then the percent cover by vegetation class, as well as a percent cover by a few other things like bare soil, leaf litter, woody debris within that 1 meter by 1 meter quadrat, times 8 quadrats. No sampling was conducted on private property. So if it was private, it was noted as private. So those plots would have 0 values for all the percent covers and would be noted 100% private. This is another example of very different from the previous one, sample plot, again, Backpack Marks Plot Center, situated in what is supposed to be a woodland, but is more of an open park habitat. The area to the left that appears to be a forest was outside the park. In terms of how this data was collected, it was collected using a few apps on my phone. This was the most convenient way for us to collect the data. If you choose to do something similar in your municipality or area, or for any project, you would not be restricted to using these apps. They're just what I found convenient, which was Avenza Maps for navigating to the points, because I could upload all the data as a KMZ file and have that all on my phone. I used Memento database, which was just a blank database to start with that I could fully customize to put in all the fields and all the data that I needed to collect for that. And then Merlin is a bird identification app that was also used to record bird calls. And then from that, I included a list at the woodland level of the birds present that were observed within the Merlin app. The current status of this project is 79 permanent sample plots were installed last summer in 2024. Sampling will resume in the spring whenever the snow melts, basically. And the vegetation starts to grow, because we do want to capture vegetation, including the herbaceous layer. There's still more analysis that can be conducted on the data that's been collected to date. However, there are some challenges with that in terms of the weighting of different attributes, and getting a representative sample of the data, given that the parameters have different values. So, for example, if it's a percent versus if it's the number of trees, you've got different scaling, which was causing some complications. The results to further continue, we had about 100 species of trees observed in the woodlands. Within the permanent sample plots, there were fewer tree species observed, about 58. Hackberry, Manitoba maple were very common across the woodlands. Fewer shrub species and tree species, anywhere from no shrub species to about 14 per woodland. Well, precisely 14 per woodland was the highest. So that's just a brief overview of the findings at the woodland level. At the plot level, we had trees. Here is a graph of the most common tree species. So we had a lot of sugar maple and Freeman maple, as well as invasive species, including buckthorn, Manitoba maple, and Norway maple were very prevalent. Most of the trees were alive and in good health. There were some dead ones. On the graph, I've got snag, which was the unrecognizable species of snags. There were some other trees that were recognizable by species that were included and noted their condition as dead. But the ones that were snags on the graph are the ones that were basically without bark or twigs left. I also classified the ecological land classification at the center of each plot, which is different from the traditional ELC methodology where you would delineate a polygon based on aerial photography in advance, and then find a plot based on a representative portion of that polygon that had been previously delineated. So this does not follow that methodology. It was done at the random plot locations. We didn't want to have 2 separate locations and different data collection for each woodland on ELC and then separately the permanent sample plots. We just wanted permanent sample plot data. So that was why we did the ELT at the plot center, and we didn't want to bias the data collection and do it the entire permanent sample plot based on a site that looked convenient or representative on the ground. We wanted randomized sampling. In terms of the ELC classifications that we found, 21% was parkland, so not woodland. That's like your manicured mowed grass, a few trees interspersed. But most of them, close to 60%, were woodland with deciduous forest. There were also some thickets, like buckthorn thickets, and a few meadows. The implications of this is that now we know for the 79 woodlands, and over the next couple of years should have data on the remaining woodlands, we know where to find different species of trees, whether that was trees counted in the permanent sample plot or trees observed within the woodland. So we have an idea of the prevalence of the tree species on a woodland basis. So what percentage of woodlands, for example, had beech, and then the percentage of trees within permanent sample plots that would be beech. So we've got two different parameters there for species diversity, which will be hopefully very helpful if any invasive species come along that we don't already have. For example, oak wilt, we know about 3.2% of trees based on current data are oaks from the permanent sample plots, and the breakdown of the red oak group to the white oak group. So that will be helpful if oak wilt arrives. Hopefully it won't, but unfortunately it's likely to come to London. We also know about 44.4% of the trees are susceptible to Asian longhorn beetle should it arrive. We know 2.6% of our trees are beech, which leaves them susceptible to beech leaf disease, beech bark disease, beech leaf mining weevil, which is not currently in Ontario, and the new Phytophthora species that was recently detected in fir, which can also affect beech. So they're quite sensitive to a number of pests, but at least we know which woodlands they're located in and approximately their size diversity as well from the permanent sample plots. So that's just to tie it all together, what the benefit of this is. And with that, I will go to questions. Thank you.
Thank you so much, Marnie. That was really great. If you have any questions for Marnie, please pop them in the Q&A box. Oh, one came in right now. Really interesting work. I'm wondering if you found any vegetative species, native or invasives, that you weren't expecting.
I found a few that I was not as familiar with, including yellow archangel, I believe it's called. So just a herbaceous ground cover that was taking over our woodland in one of the locations. I haven't found anything particularly unique, but I did find a massive serviceberry in one of the woodlands. It was probably trunk diameter about 25 to 30 centimeters, which was rather surprising. Very nice woodland it was in, very mature.
Very cool. This person says very cool, thank you. Yeah, if there's any other questions, please pop them in the Q&A. We'll also be putting Marnie's email and the other speaker's email in the chat too. But yeah, your talk was really informative. I think long-term monitoring is so important, especially for early detection and, you know, rapid response of those new types of invasives. April here is asking, just wondering if buckthorn treatment is on the plan. If so, what types?
Some of the buckthorn has been treated in the past, which was interesting to see as well, because there were cases where it had been treated and either you had no understory coming up underneath it, or you had a sea of little buckthorn babies. I believe they had done pesticide treatments, so a foliar spray had been done. I think that was the most common treatment that had been applied.
Good to know. Okay, thank you so much, Marnie. If there's any other questions, you can reach out to Marnie directly through email or through the Zoom events platform. Marnie, I love those images on your slide too. Those are really cute.
Thanks. We had a local artist draw them for us for some of our outreach.
Love it. Well, thank you so much again for joining us. I'm happy to introduce our second speaker, Cailyn Carscadden from the University of Toronto, to talk all about oak wilt. Thank you so much for joining us, Cailyn.
So sharing.
Thank you. And just give me a second while my camera on button disappears so I can share my screen. All right. Is it? Oh, it's happening. Okay. Excellent. All right. So thanks so much for having me, everybody. My name is Cailyn Carscadden, like Maddie said, and I'm a first-year master's student from the University of Toronto. At the university, I'm supervised by Dr. Sandy Smith, and then at the Ontario Ministry of Natural Resources, I'm supervised by Dr. Sharon Reed. And my research focuses on improving environmental DNA surveys for the early detection of oak wilt. So for those of us who haven't heard of it, oak wilt is a vascular disease caused by the fungus Brezziella fagicearum. And it's characterized primarily by these dark fungal pressure pads that you're seeing on the screen. as well as wilting leaves that come off the tree around July or so, but this is a symptom that's quite general of several fungal diseases. Like Marnie was alluding to, oak wilt does affect all oaks, but primarily any oak in the red oak group. So for those that aren't forestry nerds like myself, that's any red oak with a pointed lobe in its leaf. So I personally first think of the northern red oak, but other oaks like the black oak, for example, are also within this group. And if they are to contract oak wilt, they will die within 1 year. Oak wilt has two main methods of transmission. The first is below ground through root grafts, and the second is overland spread, where this disease is vectored by sap beetles in the family Nitidulidae. So below ground spread is the most common method of transmission for oak wilt, and this is the primary way that oak wilt travels to new trees within stands. So after the fungus moves through the entire vascular system of the tree, it'll then travel to a healthy tree by moving into the roots through the root grafts that connect the disease and the healthy tree. And this way, oak wilt is able to infect multiple new trees a year, and that also creates these characteristic pockets of oak wilt that you're seeing on the screen here, which can be helpful for remote sensing. What this research is much more interested in, however, is how oak wilt is vectored by sap beetles. So the year after a tree has passed away, it'll produce that same fungal pressure pad I showed you before. And that pressure pad will push a crack into the bark that allows the fruity smell of the fungus to attract multiple different species of sap beetles, which then crawl into the bark and then get covered in all these viable oak wilt spores. If these insects are then to visit a healthy tree with an injury, this has the potential to create a new infection center for this disease. In Canada, we had 3 confirmed detections of oak wilt in 2023 in Ontario. 2 of them were in Niagara County in the south here, and then another one was in Simcoe County in the north. All of these trees underwent sanitary removal, and luckily we didn't see any new oak wilt in 2024. So this is the good news, but unfortunately, if we take a look at these climate models I have on the far left, we can see that Canada does have quite a suitable climate for oak wilt, both now and in the future. And we also have quite a broad range of oak hosts. So in the climate modeling, this hatching that goes from Manitoba into the Maritimes is, I think, most of the oaks we have in Canada, and then particularly the northern red oak, Quercus rubra, which extends from, I think this is still in Ontario, all the way up to the Maritimes as well. If oak wilt does become established in Canada, the potential cost of street tree removal alone is between $226 and $420 million. And this does not include any losses to our GDP from the loss of oak timber, and it also doesn't account for any losses from the cultural and ecological values we draw from these trees. So all of this is to say that we need a robust early detection program for this disease. Early detection is the process of detecting an invasive species either before or soon after symptoms are detected on the ground. So borrowing from the schematic from our hosts here, early detection aims to extend this prevention and eradication phase so we don't have to move into long-term control. Early detection can look like citizen science as well as remote sensing, which can be helpful for this disease, which I mentioned earlier. And in recent years, particularly environmental DNA surveys. eDNA surveys can often be much more reliable than visual inspection in terrestrial systems, so they've become increasingly popular in recent years. Environmental DNA is any genetic material left by any organism in the environment. So in my case, we're particularly interested in those oak wilt spores or the oak wilt pathogen. But we might also be detecting any fragments of the mycelial mats that the insects happen to pick up with their feet when they're walking on it. Once you have an environmental DNA sample, there's quite a variety of different things you can do. Many experiments will do a detection. So do I have oak wilt or do I not, based on the concentration of DNA that I put into my reaction? But what several early detection surveys are interested in is this quantification, or qPCR. PCR stands for polymerase chain reaction. And when we quantify this, when we put our sample into the machine, through the process of the entire reaction, it'll count millions of copies of, in my case, oak wilt DNA to help inform us how much oak wilt did I have at the beginning of my reaction, and what does this tell me about how much oak wilt I have on the landscape. So previous research for using quantitative PCR for oak wilt has seen the assay, or the primers in the probe, and this is the chemistry that's necessary to identify the oak wilt DNA within the sample. In 2022, this was developed for the first time. And then in 2024, Gauthier et al., a different team, proved that this technology worked better than the previous best technology for the molecular detection of oak wilt. It was also proven that this assay could reliably detect oak wilt on insect samples. So again, this is the really good news. We have technology that's extremely specific to the oak wilt fungus that works extremely well and can detect oak wilt using the same method of transmission that it would use to come to Canada. But the drawback to using this technology right now is that we don't currently have the baseline values for how much oak wilt DNA to expect in an active stand compared to a non-active stand that would give us the ability to be able to interpret what these surveys mean in a landscape context. So the objectives of my study are to describe the spatial and temporal variation in the overland spread of the oak wilt pathogen in an active and a non-active stand. And through doing this, to describe a baseline of oak wilt DNA for Southern Ontario biosurveillance studies in the future. The second objective, which I haven't given quite as much background on, is to describe the variation in the different amounts of pathogen carried by these different species of sap beetles as vectors in active stands. And through doing this, we'll be able to improve Canada's early detection program for oak wilt. The trapping methods for this study are the same across the board. We used a black 12-unit Lindgren funnel trap that was baited with whole wheat fermenting bread dough, as well as the pheromone lures for Caplothorax sayi and Coloptris truncatus, which are two well-known vectors of oak wilt. And the collections were made every 2 weeks for this experiment, no matter what experiment they were a part of. Getting into where we put these traps, in Southern Ontario, again, we're interested in looking at what the current levels of oak wilt DNA look like here for biosurveillance. And we're also interested in describing what the spatial variation of oak wilt is in a non-active stand. So we conducted a broad-scale monitoring study by working with 19 different partners from different municipalities, conservation authorities, and different branches of the Canadian Food Inspection Agency. We only asked them to trap during the high-risk period for oak wilt spread. This is when those sap beetles are the most active, so between May 15th and July 1st. So they made 2 collections, and we only asked them to set their traps up nearby oak trees and at least 20 meters apart. And then this extremely vague list of instructions saw traps set up in quite a variety of different habitats. You're seeing 2 examples in the bottom here, but other traps were set up in city parks, along urban or nearby urban street trees, in golf courses, country clubs, and then also along county roads. So quite a variety of different spaces where we may see oak wilt in the future. In Michigan, where we do have active oak wilt, we were interested in describing the spatial and temporal variation in the overland spread of oak wilt, as well as the variation in the amount of pathogen that vectors are carrying. So for those of us familiar with the shape of the Lower Peninsula, Alpena is kind of in like the first knuckle of your first finger. So that's where this was. To set up the transect study for spatial variation, we first identified a sporulating mat, so that black mat that I showed you earlier. Here's another example in the corner. And then set up our transects, which are these orange dots. Each of these transects made 2 collections during the spatial study, which occurred during that high risk period. And then after that was done, by June 26th, we took down half of these traps to continue sampling until August 20th. And this was able to inform us how these spatial changes also vary through time. The other thing that was important to this study was because we found all of these first mats nearby oak wilt pockets, and you're seeing an example of this beside that first oak wilt mat. We also went around each transect and identified the closest sporulating mat to each trap, which would inform us the smallest distance that an insect would have to fly to carry oak wilt to these traps. For the vector study, we only set up 2 extra traps, which you're seeing underneath this northernmost transect, and collected from these twice so we could do more work in the lab for this one. So getting into what we did in the lab, for the spatial and temporal studies, we left these as bulk arthropod samples. So what this means is that we took out all of the plant material, kept all the bugs together, and then freeze-dried this for 5 days, and then ground them in liquid nitrogen, and that's what's happening in this mortar and pestle here until they reached a fine powder. For the vector study, we were really only interested in what the sap beetles were doing. So we separated those out and then identified them to species using morphological ID characteristics, kept these species groups together, froze them at -80, and then beat them before they too were at this fine powder stage, and we could do DNA extraction, which is what's being set up in the centrifuge here. And then we moved on to PCR, our detections, and quantitative PCR, our quantifications. Where we're at currently with this project is that fieldwork, the sample prep steps, and DNA extraction have been completed for all of the 2024 samples. And we've also done the PCRs for all the 2024 samples as well, but we will need to do some of this again. After this is done, we can move on to the quantifications as well as the data analysis for 2024. I do have some preliminary results to share from that first round of PCR. So in Michigan, this is a pooled data set. We have 100 total samples and 47 of them are suspect positive. There's no real spatial trend in where these positives occur, but temporally it does look like most of them are occurring in June, and 35 out of 47 of these samples were suspect positive in June. More results will or more trends will likely be revealed once we start getting into the quantifications. In Southern Ontario, we had 65 samples and 6 of them are suspect positive. And before any alarm bells start going off, most of these positives typically appear together, and they also occur in areas where we expect there to be higher levels of oak wilt DNA. So areas that have had a previous confirmed detection, as well as areas that are nearby a neighboring US county with oak wilt, are areas where we saw these. So no real surprises here, and more information will be revealed when we move into the quantification stage. For the vector study, we had 52 subsamples and 4 of them are suspect positive. 3 of them all occurred on the same date, that's June 26th, but only 2 of these 3 species have been implicated as vectors for oak wilt before. So Glystrochyllus fasciatus is this kind of lighter orange guy with like the cow-shaped spots on his wings. This one was implicated as a vector for oak wilt. First in New York in 2018, and then again in a different paper that came out of the other side of the northern peninsula or the lower peninsula of Michigan. And that same Morris et al. 2024 is the paper also implicated Glystrochyllus sanguinolentis as a vector for oak wilt from a very similar latitude in the lower peninsula of Michigan. Cryptarcha ampla is this other June 26 species. This has never been implicated as a vector before. And then the only species to test positive in July was Caplothorax sayi. But because this is a well-known vector of oak wilt, this wasn't really a surprise. The next steps for this are to optimize the PCR protocol for detection and then rerun all of our samples. We'll also be performing quantitative PCR on our samples in the coming weeks. And then once that's done, we can move on to the stats. Again, I am a first-year master's student, so I get to repeat this study in the spring of 2025, and this time have a larger temporal component funded by the Canadian Tree Fund, which will not only enable us to sample for much longer in the year, but also to sample in Ontario as well in some locations. I would like to thank the Canadian Food Inspection Agency, as well as the Ontario Ministry of Natural Resources, the Manitoba Agriculture Resource Development, U of T, the Michigan Department of Natural Resources, and the Surge International Committee for funding and other contributions made to this project. These eDNA projects can be expensive at times, but they are quite insightful. And then particularly the Michigan DNR in that other category for helping us find some of our sites. And then I'd also like to thank all of our Southern Ontario partners who were able to help us do the broad scale monitoring study. This research would not have been possible without any of these organizations or any of the last organizations on my previous slide. So thank you all very much for helping me do my research. And then any questions, I should have a few minutes to answer some now.
Awesome. Thank you so much, Cailyn. That was a really great talk. Really cool research. Oak wilt's obviously a very hot topic now and a big priority species with it recently being confirmed. So it's really cool to see the different research that's going on.
Mm-hmm.
And we do have a question that came in. Have you sequenced any of your PCR positives to confirm oak wilt?
We have started that. Unfortunately, we sequenced outside of the MNR. Unfortunately, they were having some problems with their equipment at the time. So we will be sending more samples for sequencing, but that is on my to-do list, if you will.
Awesome. What was the minimum distance you noticed the beetles traveled?
That wasn't something that we could track because we weren't doing kind of we weren't doing a mark recapture study. We have seen other papers have separated them 20 meters, others 30 meters. So we decided 40 meters was a good idea, but we don't have a current recorded longest flight distance. But we would assume that most of them would kind of stay around where there's more food, and that's where there's more oak wills.
Yeah, no, definitely. What sort of distances were your traps from those spore mats? Hundreds of feet, thousands of feet, just trying to get a sense of scale. They say very interesting work.
Oh, thank you. So I don't know my meters to feet conversions. Hopefully the person who asked that question has a calculator on hand, but our first traps were set up within 40 meters of that first oak wilt mat that we found. And then all of the traps in the Michigan study were 40 meters apart. The largest distance we saw with our trap was, I think, 160 meters. So when we were looking for more oak wilt mats, we were only looking within that kind of 160 meter radius of that last trap.
Awesome, sounds good. This person's asking if you found any differences between like an urban setting versus a more rural setting.
I'd have to take a closer look at my data to answer that question super well. The only thing that I think I do remember from the sample processing is a lot of the urban ones tended to have like more plant material because it was unobstructed by the wind, but that was common for all of our samples. But I haven't noticed too much yet, but more to come in my next year's data analysis.
Sounds good. How do you make sure you're not carrying oak wilts between sites?
So to our current knowledge, oak wilt is not airborne, and I was also not the one to do the Ontario sampling. And there's a lot of oak wilt in Michigan, so we weren't super worried about we weren't infecting any new trees because we were only looking at dead trees that have those fungal mats.
Awesome. Yeah, sounds good. I know it's a different situation in Michigan, especially compared to here in Ontario. I know when we had the oak wilt detection, when we were managing it in the city of Niagara Falls, there was a ton of precautions with a lot of, plans for who's coming to the sites when, a lot of decontamination protocols. So it's a big, extensive process, especially depending where you are.
Yeah.
Okay, yeah, thank you so much, Cailyn. If there's any other questions, you can reach out directly to Cailyn. Her email is provided in the Excel doc we put in the chat there, or you can reach out through Zoom events directly. Thank you so much. Appreciate it.
Thank you.
Our last speakers of this session are Glen Prevost and Olivo Mehdi from the Ontario Woodland Association to talk about Hemlock and their Hemlock Champion project. Take it away whenever you're ready.
All right. Thank you, Mehdi. Olivo's going to share his screen. I'll get started here. But yeah, my name's Glen Prevost. I'm the program manager for the Ontario Woodlot Association. I manage a number of different programs, and this is one of them. I'll let Olivo introduce himself before we jump in. I'm going to take the first half of the presentation, and Olivo will take the second half.
Okay, thanks, Glen. Yeah, my name's Olivo Mehdi. I'm the Hemlock Woolly Adelgid project technician with the OWA, so I got to be really specific into this. And that's why I'll cover the details after Glen goes over the intro, but it was super, super fun.
Yeah. In terms of this project, I kind of came up with the concept, but Olivo was really the one who drove it and made it all happen. So, Olivo, can you go to the next slide? So, the OWA has been involved with Hemlock Woolly Adelgid management and monitoring for the past 3 years. Recently it had entered Ontario. What we had noticed was that we didn't have some of the tools needed for an operational response. There was really good informational response, but nothing on the ground. And so we needed a solution for that. We also needed information for smaller forests because that's generally who we deal with. So we came up with this concept. We were going to treat 500 hemlock trees to learn some lessons about what application looks like, how to set up teams. We were learning from others in Nova Scotia as well. And then to develop a practical guidebook, something that a practitioner or a knowledgeable landowner could pick up, read and then be able to set up their own plan. It gets into pretty significant detail. And the goal is to expand treatment of hemlock woolly adelgid generally by pesticides. Next one. So our project where we treated the trees was Long Point Region Conservation Authority lands, and we owe a great debt to Deb Thane and Chris Reinhardt, who are there on the left. Without them, I can safely say that this wouldn't have happened. Same with Trees Unlimited and Paul Robertson and Reed Robertson, who were consultants that helped us, in addition to Olivo and our own staff on site. They're treating on private land right now, so they bring some pretty significant tree marking experience to the project. And then finally, we funded this project with an invasive species action fund grant in the transformative action stream. So I think it was a good investment of money. And then also we had project learning tree funding to cover a significant portion of Olivo's time. So I'll turn it over to Olivo to talk about what it is that we actually did, but I was just trying to set the context there.
Thanks, Glen. So like you said, we developed this guidebook, and it was a pretty like multi-step process. But the first thing, of course, was to research the up-to-date practices. So what the states is doing, they have a huge infestation for many more years than Canada has had. Some information out of Nova Scotia, a little bit of things out of Ontario, some reports more than research. Then through all that reading and talking, you compile a list of equipment and methods. So there's a pretty big, extensive, very detailed list. And of course, if you miss something tiny, it might throw you off for a whole day. So it was really key to get that all together and then to be able to write that down for other people. I met with practitioners who have performed treatments, Zoom, online, emails, in person. One good visit was Royal Botanical Gardens, where they have had a pretty active infestation for a few years, but it's only been identified recently, and they've treated, so you got to go through that procedure with them and see it in person rather than just on a computer screen. Acquire the equipment, so ordering things online, borrowing things from people once we made that list, buying things, double checking, going to Canadian Tire, all kinds of trips, treating an at-risk stand of hemlock, and that's the practical portion. So then using the information we got from treating it ourselves, what worked, what didn't, to kind of polish the guide and make it very usable and not leading you into errors. So there's 3 management response options that we focused on. There's silviculture, and that's to improve the resilience of the forest stand and make it less habitable for HWA. But it's good to note that that doesn't stop anything. It won't prevent mortality and it won't stop spread. It will just potentially boost the health of trees so that they're more resilient and you have a longer window to work with. But this is currently available to anyone in Ontario. Then there's the insecticides, and that reduces the active infestation and could prevent ones in the future. So it's the only way we have right now of preventing mortality, and it's currently available, or at least reducing the percentage of mortality. And then finally, there's biocontrol, which is the release of predatory beetles and flies that feed on the hemlock woolly adelgid. But we cannot use it in Canada. It's being researched in US and Nova Scotia. And I guess you could say it's like the main hope for a long-term landscape control of hemlock woolly adelgid, because it's just not practical to inject or spray every single tree that you want to protect. So in the very beginning, this very, very helpful meeting that kind of kickstarted everything, Sinead Eget and Ashley McRae of the OWA went to Nova Scotia to work with Donna Crossland, who's there in the middle of the left picture. And she kind of can be seen as spearheading the treatment effort in Nova Scotia because they got the infestation a lot more in advance than Ontario or other provinces did. And she's really developed her own methods and own training, and it's what we kind of use to inspire our own. And she trained up our own helpers, and they got me to up to date on everything and brought really useful information. So there on the right, you can see like some of the equipment they're using as a loading for injection, but we'll get into that as well. So the actual applications that we did, in general, the current most effective management option is for saving trees is this pesticides. Like I said, the goal is to lifeboat stands until the biocontrol becomes available, and there's no real timeline for that. It depends on many, many things, including climate and government, and even the survival rates of insects, right? But it's lifeboating as in just stopping every single tree from dying, trying to keep genetic diversity and stand function. So all the available pesticides, they're systemic and they're applied either as a basal bark spray or a stem injection, and they get transported into the canopy through the vascular tissue. And then when the HWA feeds, it's a toxin that kills them, and it's pretty specific, like it targets them exactly, rather than other things interacting with the tree. But you need an exterminator license in Ontario to purchase any of these to use on your own land. And then you need very robust insurance to apply it as a contractor on any land other than your own. And that's when you receive payment or some form of compensation for doing it. And then you also need an operator license. So it's multi-step. But we focused on these available pesticides, Zytect 2F, Starkle 20SG, which are both basal bark sprays, and Imajet 5, which is an injection. And the two sprays, they're only emergency registration, so they may expire. It means they're just temporarily allowed for use in Ontario, but the injection has a full registration. So on the left here, there's a photo of filling prep for tree injections. So these are canisters we fill via like a compressor. And then they get plugged into nozzles that are in holes in the trees, like a reverse maple tap almost, just low to the ground. And then on the right, there's the basal bark spray happening. And this would have been Zytect 2F. So there's a summary of the treatments. I won't go through everything, but there's some pros and cons. The sprays that I mentioned, they can't be applied within 7 meters of any surface water or water that you consider life supporting, because you want to prevent drift and toxicity to any aquatic animals. So that we call it the 7 meter buffer that comes out of Nova Scotia, but it's been applied to the Ontario labels. And then there's a limited amount per hectare. So in this product and DBH limit column of the table, you see you can either look at it in centimeters of DBH or product volume, but you're limited in that. So then you can do it the next year to treat the rest of the trees. But the injection is time consuming and more costly. It can be used to protect the most valuable hemlock near water because that's really the niche function of hemlock. And then there's, of course, an unlimited amount per hectare, so you can treat as many as you can physically with the time or money that you have. Starkle spray acts rapidly but wears off quickly, so it protects within the same month you treat it, but by the next season, it will probably be not protecting anymore. And then Zytect and Imajet, they take longer, but they protect the trees for a very long time. And then there's just a note of the alternative option. It's a more organically sourced. It's neem seed pod oil, I think, is the main ingredient, but it's the most expensive and it doesn't last very long, but it acts very rapidly. I think I need to speed up a bit. So the prep work we did in September, Long Point Regional Conservation Authority staff measured a bunch of hemlock trees and marked them all, mapped them, took down their DBHs for us, and that was about 1,700 trees in this one tract. So then we came in with Trees Unlimited, and we selected 605 high priority trees for treatment, measured them, flagged them with the color code telling us which treatment we're going to use. That took 3 days with the crew of 4. And that prioritization is a function of the tree's health, sometimes the DBH. So you pick not only the biggest trees, but also a range of DBHs at the healthiest you can, just to ensure they're most likely to survive. So there's the map of those 1,700 trees on the left in this 16-hectare plot. And on the right, all the hemlock trees fell within 6.5 hectares, and we divided it up into 1-hectare blocks to stay within our limits. Okay, so the prep for the treatment after the selection, we summed up the DBH data by chemical and by hectare, and then you find out how much chemical you need just by doing some simple multiplication. Trees that were over limit, because even though we split it up into hectares, we might have marked too many trees in some areas. We had to remove just to stay within our hectare limits. So we wrote a list down and just ignored them day of. Repaired a leaking EcoJect system after trial run, so that's the injection manifold. Needed some repairs because it was a lend from a different agency, but the Starkle emergency registration did not get renewed, so we treated it in its like little dead window when it was not legal for use in Ontario, but now it's been renewed again. And it's good to know that the insurance took a very, very long time because of how unique it is, and it delayed the treatment a lot. So if it's ever the case where a contractor or an operator needs the insurance, they should begin months in advance, at least for now, before that comes more mainstream. So there's an injection happening around the base, and these pressurized canisters push it into the xylem. And then here's a spray happening like we showed earlier. This is again Zytec, kind of chalky once it's diluted. Setup for the injection, you need a generator to run the compressor to fill the canisters, drill charging station for drilling all those holes. But if you were either filling the canisters not in the field, you could just bring them all with you. You wouldn't need this whole setup. Or if you were just spraying, you wouldn't need this whole setup. Just the safety gear that's in the back, the PPE, eye wash station, garbage, that kind of stuff. So that work that actually happened for the treatment, it happened in October 2024, delayed again because of the insurance, but we were very lucky that the weather was so warm in the fall. I mean, not so good for other things maybe, but just to get this treatment in. Took 3 days with a crew of 6. 3 people injected, 3 people sprayed, but the spraying is much faster than the injection, so they finished in a day and a half, and then they were able to help us. And of the 605 trees we selected, we treated 530, 21 partially treated. That means at the very last day, the weather turned, it got cloudy and windy, and there was absolutely no uptake for about 4 or 5 hours. And because of insurance, you can't leave them there, so you have to pull them unless you're on site. So eventually we took them out. And then 54 of those over limit trees didn't get treated. We used about equal amounts of Imajet 5 and Zytec, so that's spray and injection. And it's what we calculated, like it was on the nose. So that was 1,235 injection sites. And that's why you need the drill batteries, because you're just drilling the base of the roots or the trunk all day. So treatment map, this was made by Deb, really nice from Long Point. So here's it say orange, that's our sprays, purples, injections. Then there's a few colors along the top. You can see the kind of turquoise. Those are the partially treated trees along this one vernal pool. And then there's some errors because we had so many trees, and that's part of what we learned. Less people, better for keeping track of the trees better. And then there are some omitted ones, like over the limit, or we couldn't find them. So we did learn lots of lessons, but insurance can take some time to obtain. Plan ahead. Think about your data management to reduce errors or your work division. The trees should be marked ahead of time to know chemical needed, and this marking takes longer than normal tree marking. There's more decision making involved. Injections take much more time than basal bark applications. And if all trees cannot be treated in 1 year, you can do the rest in year 2. Plan well ahead again. Of course, emphasis on that. Everyone knows. But right crew size makes work efficient. So it depends on the work you're doing, the experience of the people, and how big the tract is. But there's an optimum amount for everything. And teamwork and partnership are key, and that's really what got us through all the stages of this. Next steps, we want to treat untreated trees at the Long Point Conservation Authority in that tract. So even if we don't do all 1,700, there's still double at least that we can do. Educate people about the guide, get it in use. It's ongoing. Educate landowners on management options for Hemlock. That's what the guide is going to achieve. And then work with CFS and CFIA to establish biocontrol in Ontario. And then expand the treatment program to treat more trees, especially high quality individuals and stands. And that will help us boost or promote genetic diversity of the surviving trees. So thanks again to the people who we collaborated with and got this going. This Debbie Thane, Chris Reinhardt, and Judy Maxwell from Long Point, Paul Robertson and Reed Robertson, and Tom Stanton from Trees Unlimited, who helped us get all the work done. Donna Crossland in Nova Scotia from Healthy Forest Consulting for the training, the ISC, and the Project Learning Tree. and then Glenn and I can answer anything.
Thank you so much, Olivio and Glenn. That was really great. I'm really excited for that guide. I think it'll be a valuable tool. I know there's so much information out there about hemlocks and management options, so it'll be good to have a guide that kind of brings it all into one place for people.
Yeah, we think so for sure.
Yeah, we do have some questions coming in. Let's see here. Are you able to touch on the specific silvicultural strategies? You referred to silvicultural briefly.
Glenn, go ahead.
Yeah. So there's a really great guide that was published last year that I would refer you to. But generally speaking, we want to increase the health of residual hemlock trees. We also want to consider the health of the remaining trees that are there. So if you are going to experience mortality, consider making sure that you have resilient trees there. But what I would do is I would refer you to the silviculture guide. That was Ken Elliott and some folks at the Canadian Forest Service wrote. There's a really good summary of that.
Yeah, definitely. Thank you. Some people are curious about the insurance. Have you found that the high level of insurance required to be able to perform injections reduces the willingness of arborists to offer this service?
I don't know if we're necessarily in a position to judge what the impact of that is. I can just say that for us, it took a long time to get the approval for the insurance. We did get it. It just took a long time, and the insurance companies were doing their due diligence. So, and it needs to be renewed every year. So it would be a deterrent if you weren't doing this on a regular basis. But this is the same sort of insurance that you would need, not just for hemlock woolly adelgid and those chemicals, but any other sort of chemicals. So if you've got the appropriate license already, you can go out and start using these products as well.
Yeah, it's outlined in Ontario regulations. I forget the exact number, but it's nothing specific to this. It's just for any operator.
Awesome. Good to know. Thank you. Does the basil bark include coloring to ensure no trees are missed during the applications, or is it the chalky look like you mentioned?
The one is clear. The one we used, the chalky one, you can see it, but eventually it dries up. I think it's better that there's no dye or anything in it, because in like a public tract or something that people access, you're not coloring trees, but you just have to keep track of what you've treated yourself. Yeah.
And I mean, there's different ways of doing that. If we flagged every tree, and there's information on those flags. So that's another way, is after treatment you could simply dot it or flag it or do something. There's a bunch of different methods, and we describe those in the guide, but it's whatever suits the site the best.
Okay, awesome. And where can this guide be found?
Well, it should be on the Ontario Woodlot Association website shortly. It's just in the final stages of external peer review. So hopefully by the end of March, we'll have the final thing done.
Perfect. Awesome. Well, thank you both so much. Really appreciate your time and your presentation. It was really valuable knowledge. I appreciate it.
Okay, thanks
All right. Thanks for having us.
Anytime. Thank you. So we'll bring this session to a close now. I just wanted to highlight that we have a lot of great free workshops coming up that you can learn more about on the Invasive Species Center's events page. Next week, actually on February 26th, we have a forest pest workshop in Hamilton we'll be talking about hemlock woolly adelgid there, and we'll be going to Royal Botanical Gardens to see it in person. So that's part of that workshop. And then on the 27th, we have an emerald ash borer workshop that'll talk about emerald ash borer and black ash, and that's in Sudbury. And both of those are completely free, so you can register on our website to save your seat. So we will sign off now, and we'll see you again at 11 for our invasive land animal session. Thank you so much.